In this, our last online lecture of the quarter, we'll learn about disaccharides and polysaccharides. In our previous lecture, we learned that monosaccharides are really just simple sugars, and we learned about their structures. Not surprisingly, disaccharides are really just two monosaccharides bonded together. What they usually look like and how they spend most of their time is just as two rings with a connection between them. And while you don't need to worry about the structure of this particular disaccharide at the moment, notice that the two rings are fairly easy to recognize. It's important to know that disaccharides exist in many different forms. Some disaccharides have two identical monosaccharides bonded together. For example, this particular structure happens to be two monosaccharides of D-glucose that are bonded together. But there's many examples of disaccharides where the individual monosaccharides are different. For example, as we'll see in a few minutes, lactose is an example of a disaccharide that's comprised of D-galactose and D-glucose. In addition to what the individual monosaccharides are, the other very important part of the disaccharide structure is the bond. And it'll matter both where the bond is located on the individual rings and how the bond is oriented, as we'll see in a moment. This special type of bond is what's called a glycosidic bond, and that particular phrase tends to be used in the context of biochemistry and the health sciences. However, this bond is actually a type of bond we've seen before. So what I'd like to do is imagine having just these two general structures without all of the additional details of the substituents. Let's talk about this glycosidic bond in more detail. What the glycosidic bond actually is essentially an acetyl bond. So disaccharides are actually acetyl molecules. We can recognize that it's part of an acetyl structure if we remember that carbon number 1, which is the anomeric carbon, is actually part of a hemiacetal. And we know that when a hemiacetal bonds with another alcohol, we get an acetyl structure. So in this case, the hemiacetal is actually the monosaccharide on the left, and the alcohol structure is actually from the OH group on the monosaccharide to the right. This particular glycosidic bond is an example of one of the most common. It's what's called a 1,4 link, or a 1,4 bond. That's because it's connected from the carbon number 1 on the left side to the carbon number 4 on the right side. If we think about this bond in more detail, all that's really happened to form it is the OH that was present on the anomeric carbon, that was part of the hemiacetal, has bonded with the OH that was on the 4th carbon from the other monosaccharide. Through a dehydration reaction, we remove H2O, which means we remove an OH from one side and an H from the other. So the process in general links together the two OHs, creating this new acetyl link, which is also called a glycosidic bond. Now, as I mentioned, the orientation of these bonds is crucial. This particular example, it was very, very easy to connect those OHs together. I essentially just moved them closer to each other and drew it as a bond through an oxygen. That was easy because both of the OHs were pointing down. You cannot change the orientation of the OHs because, as we saw previously, that would change the identity of the actual glucose molecule. So instead, what we need to do is reorient the molecules. Let's look at how. In the example we just saw, it was relatively easy to deal with the orientation of the bonds because both of the OH groups were already oriented pointing down. In the case of the monosaccharide on the left, this OH group is pointing down because whatever monosaccharide it must be has the OH in the alpha position, pointing down. The OH group on the right-hand molecule is pointing down because it must be part of a monosaccharide that has that particular orientation of the OH groups. In either case, these OHs must stay in the orientation they have. So to make the glycosidic bond, I simply brought them together, essentially removing a water molecule to complete the link through a single oxygen atom. We've already said that this is what's called a 1,4 link because the link takes place between the carbon on the left, which is carbon number 1, and carbon number 4 on the right-hand molecule. More specifically, we would actually call this an alpha 1,4 link, or an alpha 1,4 glycosidic bond, because the carbon number 1 on the left-hand molecule has the OH in the alpha position. Now let's think about how this would be different if I was dealing with an OH in a slightly different orientation. In this case, my monosaccharide on the left-hand side still has an OH in the alpha orientation. However, if I were to bond this particular monosaccharide to another monosaccharide in which the OH was oriented up, now it's not quite as simple because I can't just swing the OH down. So let's think about how we would have to deal with a glycosidic bond between these two molecules. Because the OHs have to stay in the current orientation, in this case the OH pointing down on the left-hand molecule and the OH pointing up on the right-hand molecule, I have to instead think about moving the molecules themselves. So in order to create a link between these two OH groups, I actually need to move the structures so that they're in a position that allows the bond to take place. In this case, rather than having the two monosaccharide rings next to each other, I would have to orient them so they're almost like a set of steps. One has to be higher than the other to allow the two bonds to stay in their relative up or down positions. This is a common issue that we run into with glycosidic bonds, and it affects how the various monosaccharides form into disaccharides, and as we'll see, furthermore form into polysaccharides. Now what's important to note is the example on the bottom is still an alpha 1,4 link, because both in the top example and the bottom example, the monosaccharide on the left had the OH group in the down or alpha position. And in both cases, it bonded to an OH group that was pointing either up or down on the fourth carbon on the other monosaccharide. So calling something an alpha 1,4 link actually doesn't tell you anything about the orientation of the bond in terms of the second monosaccharide. However, we always have to maintain correct orientation to ensure we are showing the true structure as it would exist in nature. This is also true when dealing with bonds in the beta orientation. So if we have two monosaccharides and the monosaccharide on the left has an OH in the beta position, sometimes the orientations are easy to deal with and sometimes we have to readjust where we would position the molecules. Because the beta position for an OH group means that the OH group is pointing up from the anomeric carbon, it's very easy to make a bond if the OH group on the fourth carbon on the other molecule is also pointing up. It's analogous to what we saw in the previous example when both of the OH groups pointed down. We simply bring them together to create the glycosidic bond. In this case, this is still a 1,4 link, but now because we dealt with a carbon that had an OH group in the beta orientation, this would be called a beta 1,4 link. And likewise, if we want to maintain that beta orientation, but the OH group on the second molecule points down, I would have to orient these molecules again, almost like a set of steps, in order to give them the correct structure so that we could form this beta 1,4 link. Now, why does this matter? Well, the fact is, even though these are chemically similar bonds, they have different orientations and different geometries. And the issue with this is when we run into glycosidic bonds, let's say in the human body, many of them go through chemical reactions with enzymes. And those enzymes are only able to operate on very specific geometries. So what that means is an enzyme may be able to break apart or work on molecules that have one orientation, but the exact same link in a different orientation means that now the enzyme won't work. And we'll actually see a couple of examples of that when we get to polysaccharides in a few minutes. Let's try a practice problem. Maltose is a disaccharide that consists of 2 units of alpha-D-glucose. Draw a unit of maltose with an alpha 1,4 glycosidic link. As you do this, I'll give you the structure of D-glucose. But we know D-glucose can exist in both an alpha and a beta form, and maltose only works with the alpha form. So here's a unit of D-glucose. I've drawn the anomeric carbon's OH group with a squiggly line, which means it could go up or it could go down. Based on your knowledge of alpha and beta, you should know which way it should really go. So once you've determined that, pause the video and draw a unit of maltose with an alpha 1,4 glycosidic link. So maltose happens to be comprised of two identical units of alpha-D-glucose. We know what the structure of D-glucose looks like, and because it's in its alpha conformation, that means that this OH group will be pointing down. So I want to make sure that that particular bond stays oriented down, whether it has an OH group on it or whether it's part of the glycosidic bond. Now to turn this into a disaccharide, I'm going to need to draw two monosaccharides. Let's start by drawing just one and then filling in the various substituents. Here's the backbone of this particular structure. Now I want to keep the OH's bond pointing down on carbon number 1, but I know that's going to become part of my disaccharide's glycosidic link. So I'm going to leave it as just an oxygen rather than putting in an OH. The remaining OH groups all stay in their appropriate orientation, in this case down, up, and back down. Now I've drawn one half of the disaccharide, but I have to decide whether to point the bond back up from this oxygen or to point it down. To know how to do that, I have to look at what happens on the fourth carbon, or rather the OH group on the fourth carbon of the glucose molecule. The OH group on a D-glucose cyclic structure is pointing down from the fourth carbon. That means that I also want this bond to be pointing down from the fourth carbon. So when I fill in the remaining backbone, it looks like this. I still have the down orientation on the fourth carbon. Everything else remains the same. So in this case, it means on the remaining carbons, my OH groups are in the same position, and on the carbon that is the anomeric carbon, or carbon number 1, because there's no glycosidic bond here, I draw the OH in the down position. Again, it's not necessary to do any fancy shading, although some people find that it looks a little bit nicer and sometimes can be a little bit easier to interpret, but it's certainly not required. So this is a structure of maltose. It's comprised of two alpha-D-glucose molecules that have come together through a glycosidic link. Now maltose was an example of a disaccharide that had an alpha 1,4 glycosidic link. And 1,4 links, as I mentioned, are very common, whether they're alpha or beta. It's also common, however, to run into links between the first and the sixth carbon or the first and the second carbon. And there could be others as well. There's a couple reasons these differences in where the glycosidic link forms can have interesting consequences for the molecule. One is simply that it ends up influencing the larger structure, and this is especially true when we move from disaccharides to polysaccharides, because if we link the molecules together in a slightly different position, it tends to influence the overall polymer-like structure. Another reason this matters is what happens with the chemical reactivity. Typically, most glycosidic links involve one anomeric carbon's OH group and one OH group that's on a non-anomeric carbon. That means that the two ring structures, at least one of them gets left open to continue reacting back and forth from a cyclic to an open chain form. That's not always the case, however, and that's something that you'll run into if you get involved in any field where you have to understand more about how we metabolize different disaccharides. Just so you know, there are some other more technical terms that get employed, especially when dealing with these cyclic structures coming together to make disaccharides. While I won't require you to know this, it's helpful to know that sometimes people refer to six-membered sugar rings, like the ones we just saw, as pyranose structures, and five-membered rings as furanose structures. And that can show up again if you go into a field where you deal a lot with metabolism and biochemistry. Let's look at a few common examples of disaccharides that you've probably heard of. The one that we did just a few slides ago was the disaccharide called maltose. And maltose consists of two molecules of alpha-D-glucose that are chemically bonded together. Maltose exists naturally in many different types of grains, and the concentration of maltose actually increases when those grains are mixed with water and left to rest. This is actually an important step in the production of some alcoholic beverages, especially those like whiskey, because in this process, often called the malting process, the grain's sugar concentration increases, which means later on they can be fermented to produce more alcohol. So the malting process is actually just a natural process that increases the concentration of maltose sugar in grains. Because maltose is a sugar and it is sweet on the human tongue, it can be used as a sweetener. However, it's far milder, far less sweet than sugars such as sucrose or fructose. However, it is still used occasionally as a food additive, not just for its sweet taste, but also for its very specific taste. If you've ever had a chocolate malt or a chocolate malted milkshake, you've had a chocolate milkshake that had maltose sugar added to it. Another example of a disaccharide you've probably heard of is lactose, or milk sugar. Lactose consists of a unit of beta-D-galactose that's chemically bonded to a unit of beta-D-glucose. It is a disaccharide, and it makes up about 5% of the weight of most milk, and it's also found in other dairy products. Now, most mammals consume milk as infants, and they're able to metabolize the lactose in that milk because we naturally have an enzyme called lactase in our digestive tracts. That goes for all mammals. However, most mammals stop drinking milk as they leave infancy. The major exception, of course, is the human race. Many human beings continue to consume milk and other dairy products even as they age. However, not all of us maintain a sufficient concentration of the lactase enzyme to allow us to break down the lactose. And if you're one of the people that doesn't have a sufficient concentration of lactase, the result is what's called lactose intolerance. It basically means that when you consume milk products, the lactose is not able to be broken down by the body, and it results in some uncomfortable side effects. Interestingly, whether or not a person experiences lactose intolerance as they get older depends on a lot of factors, but one of them is actually your genetic and ethnic background. Certain genetic backgrounds can make you more or less susceptible to lactose intolerant. And in places where an entire cohort or group of people may have very low concentrations of lactase enzyme, we tend to see that those cultures tend to shy away from dairy products. One of the most classic examples is the country of Japan. People who are ethnically Japanese tend to have lower concentrations of the lactase enzyme in their digestive tract as adults. Consequently, in Japan, there's very little dairy product that's consumed by adults. Very different from other areas of the world where dairy is a very important part of the adult diet. Now let me mention one other disaccharide that you've inevitably heard of, which is sucrose, better known as table sugar. Sucrose consists of a molecule of alpha-D-glucose chemically bonded to a molecule of beta-D-fructose. And what makes sucrose interesting chemically is that it has a 1,2 anomeric link. It's a link between the anomeric carbon on the glucose molecule and the anomeric carbon on the fructose molecule. Because both of the anomeric carbons are chemically bonded together through an acetyl link, sucrose is far less chemically reactive than some other disaccharides. And if you go on to study anything in the health sciences, or especially nutrition and diet science, that becomes very important to understand. So these are just 3 examples of the many different disaccharides that are out there. Now let's move on to see what happens when we combine not 2 monosaccharides together, but rather hundreds and thousands of monosaccharides. The result is what's called a polysaccharide. And like the name implies, polysaccharides are really just polymers comprised of monosaccharides. A typical polysaccharide will have one monosaccharide unit that repeats thousands, even hundreds of thousands of times. The bonding in these polysaccharides is just as important as what the individual monosaccharide units are, just like what we saw with disaccharides. And because this is such an important concept, we're going to focus on it specifically as it applies to glucose polysaccharides. So there are many other polysaccharides out there, but we're going to focus on the glucose polysaccharides specifically. We already know what a monosaccharide cyclic structure of glucose looks like. For example, here's an alpha-D-glucose monomer. When they chemically bond together, however, rather than getting this simple disaccharide 2-ring structure, we get much larger structures, and those structures can have quite a bit of variability, both in how they link together as well as how they branch and how they behave. So let's look at some examples of glucose polysaccharides. The first example I want to show you is called cellulose. And cellulose is a glucose polysaccharide that makes up the cell wall in plant life. Now, when we think about cells, we usually think about very, very small objects. But if you have billions of those objects, each one surrounded by a very small rigid wall, it ends up giving structure to the plant in general. And in fact, up to 60% of a plant might be cellulose, really just a glucose polysaccharide. That cellulose has enough structure that it allows the plant to fight gravity and grow up and off of the soil. The monomer for cellulose itself is really just a beta-D-glucose molecule. So again, it's D-glucose with the anomeric carbon's OH group in the upward pointing beta conformation. What's really significant, however, is that this means when the molecules of glucose are bonded together, we're dealing with a beta-1,4 link. And that means we have to align the molecules to allow the upward pointing OH group on the first carbon to bond with the downward pointing OH group on the fourth carbon. Now, don't worry too much about being able to picture this, but just trust me when I tell you that when this happens through thousands and thousands and thousands of beta-D-glucose monomers, the result is a structure that has a massive ability to hydrogen bond with itself. And that's really where the ability of plants to have such rigid structure comes from. So if we were to actually picture these molecules in 3 dimensions, there would be a lot more to consider about bond angles, intermolecular forces, hydrogen bonding, and so on. But the end result is that all plants are able to maintain this rigid structure comprised of a cellulose polysaccharide. Now, the only thing I really want you to worry about when it comes to cellulose structure is what happens because the cellulose has beta-1,4 links. Even though that does affect the molecule's physical structure, it also affects its chemical reactivity. And the most important way it does that is it limits whether or not this molecule can be broken down by certain organisms. In order to digest cellulose, just like digesting any other polysaccharide or disaccharide, you would have to have an enzyme that's capable of breaking that beta-1,4 link. And unfortunately, you, as a human being and a mammal, you do not have this enzyme. The enzyme that's able to break apart the beta-1,4 link is generally only found in species such as termites and bacteria and other microorganisms. So what this means is when you consume cellulose as a human being, your body's not able to break apart the individual molecules, and therefore it can't access any of that glucose. Some mammals have managed to get around this using a symbiotic relationship. And the classic example is that most cows actually have microorganisms in their stomach that are able to break down that bond. That means that cows, while they wouldn't on their own be able to consume cellulose, they are able to break down plant life, for instance the grass they may be chewing on in a field, and chemically use that glucose for energy. The only other species that can do that are things like termites and bacteria, which is one reason that you can't survive on wood if you were lost in a forest, but termites are perfectly happy surviving on the wood that makes up your house. So, the beta-1,4 link that combines glucose molecules together to make cellulose is something that human beings cannot metabolize. Starch is an example of a glucose polysaccharide that human beings can metabolize. And what's so interesting about this is that starch and cellulose are incredibly similar. First of all, cellulose and starch are both produced by plants. Cellulose is produced by plants to give them structure, whereas starch is produced by plants as a form of energy storage. It's essentially how they store energy from the sun. Another thing cellulose and starch have in common is they both have the same monomers. Their monomers are essentially a molecule of D-glucose. But the major difference is whereas cellulose has D-glucose monomers in the beta arrangement, starch has D-glucose monomers in the alpha arrangement. So the OH group on the first carbon is pointing down. That means when you link multiple D-glucose monomers together, they'll be linked through an alpha-1,4 link. And that means you have a totally different macrostructure. In fact, if you just link 2 alpha D-glucose monomers together, what you have is maltose, which we've already seen. That's a disaccharide. But if you were to take this D-glucose monomer and repeat it over and over and over, you would get a much longer structure. And again, while it can be difficult to visualize these in 3-dimension, the most important thing to know by far is that this means your body can break down the starch because your body contains enzymes that are capable of breaking an alpha-1,4 link. So again, remember, cellulose and starch both come from plants and are both comprised of D-glucose monomers, but cellulose has an undigestible beta-1,4 link, whereas starch has a fully digestible alpha-1,4 link. Now there's actually 2 different types of starch. The first is what's called amylose. And amylose is what happens when you bond the D-glucose molecules together through alpha-1,4 links, and you get relatively short chains that sort of curl or spiral around each other. They can still contain hundreds of different monomers, but they're relatively short compared to some of the larger polysaccharides that are out there. About 20% of starch is amylose, and what's really significant about it is that it is water soluble. Now there's a classic demonstration of amylose that you can try at home that involves nothing more than your mouth and a piece of dried uncooked pasta. Pasta contains almost entirely starch, and about 20% of that starch is amylose. When you place a piece of dried uncooked pasta in your mouth and just let it sit there, that amylose starts to slowly dissolve into your saliva. Your saliva, in turn, contains the enzyme that's required to break the alpha-1,4 link between those different glucose monomers. So as you sit there, probably feeling kind of silly with this dried uncooked pasta in your mouth, your body actually starts to metabolize the amylose. And the consequence of this is that after about a minute of it sitting in your mouth, it will taste very, very slightly sweet. That's the glucose monomers becoming unlinked from the amylose chains. The other 80% of starch is what's called amylopectin, and it's a much longer, more complex network of glucose monomers, basically because the chains become cross-linked. Those alpha-1,4 links still exist, but now we have links that connect carbon number 1 to carbon number 6 or some other carbon. Those cross-linked chains are far more rigid, and while they make up 80% of starch, they're not water soluble. Basically, the water molecules can't do a good job of surrounding that cross-linked structure. So while you take a piece of pasta and you put it in your mouth, up to 20% of it might dissolve in the water, but 80% of it is this amylopectin that won't. And that's why when you soak pasta in boiling water to cook it, some amount of it dissolves into the water and you get that starchy, cloudy pasta water, which is actually really good for cooking in and of itself. But the other 80% of the pasta does not dissolve. It doesn't just fall apart. That's because not all of the starch is water soluble, and that's because a large amount of it is amylopectin. So to summarize, starch, whether it's amylose or amylopectin, is how plants store energy. And in turn, human beings are able to consume starch, whether it's amylose or amylopectin, and use their enzymes to break the alpha-1,4 links to get access to the glucose monomers. Even though amylopectin won't dissolve in water, your human body can still metabolize it. It just doesn't happen as easily as amylose, which dissolves in your mouth. Now, starch is how plants store energy, but how do human beings store energy? Well, there's actually multiple ways that human beings are able to store energy, and it's something you'll go into in a lot more detail if you go on to Chemistry 123. You'll learn a lot more about fat, which is really fatty acid esters, also known as triglycerides. Fat is a way that human beings store energy that has a very high energy density. For every gram of fat in your body, you could release about 9 kilocalories of energy. The downside to this type of fat storage is that that fat is relatively slow to access. So as anybody who's ever tried to diet knows, usually losing fat is the last step in dieting. Everything else goes first. Another way that human beings are able to store energy, however, is much in the same way that plants store energy. It's using what's called glycogen. Glycogen is a very starch-like polymer that's comprised of D-glucose monomers. On a physical level, there's just a slightly different pattern to the branching in glycogen than there is in starch. So while plants can produce starch and store it, human beings can produce and store their own glycogen. And glycogen doesn't have quite the energy density of fat, but the advantage to it is that it's far easier to access. Now, a good example of this is if you've ever participated in our Bloomsday 12K run. That's about a 7.5-mile race, or a walk for some people, that winds through downtown Spokane. If you go out and get involved in that particular activity, you might eventually run out of energy, probably at the bottom of the very large hill on the course called Doomsday Hill. At that point, some people say you hit the wall. It means you've just run out of energy and you don't want to go any further. You might have experienced this yourself, whether we're talking about going for a walk at Bloomsday or whether we're talking about studying for an exam. Hitting the wall, biochemically speaking, means your blood glucose level has dropped to a point where your body doesn't want to function anymore. But it's not the end of the road, because you have a certain amount of glycogen stored in your body that you can access. So you can access the glycogen, which is usually called breaking through the wall, and you get your second wind. The glycogen is stored throughout your body, but especially in places like your muscles and your liver. And your body can get better at accessing it with better training. So in addition to doing things like improving your aerobic capacity and building muscles, when you train for an event like Bloomsday, just by going out and walking well before it repeatedly, you're training your body to access the glycogen faster. So you're less likely to, quote unquote, hit the wall. Now, if you were to go out and walk Bloomsday every day, or maybe walk a lot further than that, at some point there wouldn't be enough glycogen, and as a result your body would start to burn fat instead. But it only does that after a lot more processes have to happen, because fat is sort of the last store of energy for the human body. Again, you'll see a lot more of this in Chem 123. But all I want you to know for this class is that glycogen is very similar to starch, except that starch is how plants store energy, and glycogen is one of the ways that human beings store energy. Now, there are many different variations on this carbohydrate theme, meaning there's many different molecules out there that are very similar to polysaccharides or disaccharides or even monosaccharides that only have very subtle differences. And I'm just going to mention some of them now because many of these are biomolecules that you've probably heard of before and will certainly hear about in the future. Many of them are based on a modified glucose monomer. So if we think about the structure of the glucose monomer, as we saw it in molecules like starch and cellulose, we know that its structure consists of several different OH groups chemically bonded to a carbon backbone. Now, if I modify glucose very slightly by changing one of those OH groups to an NH2 group, I get a different molecule, but it's one you probably have still heard of. Do you recognize this molecule? Well, what I did is I added an NH2 group. Do you remember what we call an NH2 group when we're dealing with it as a substituent? We know nitrogen is usually part of amines, so this could be called an amino substituent. And in fact, what you're looking at here is the structure of glucosamine. It's really glucose with an amine functional group added to it. Now, I bring up glucosamine in particular because it's essentially the building block of some very interesting biomolecules. If you take glucosamine and add an acetyl group to the nitrogen, you'd have N-acetylglucosamine. And again, if you take that structure and turn it into what's essentially a polysaccharide, you get some biomolecules you may have heard of. One of them is called chitin. Chitin is the building block for most of the shells and exoskeletons of all the species in the world. That means everything from insects to crustaceans to arthropods have chitin, which is really an N-acetylglucosamine polymer molecule. Your synovial fluid in your joints is also a polymer molecule that's built off of a structure that starts with N-acetylglucosamine. And chondroitin, which you may have also heard of in relation to our joints because it's part of our cartilage and connective tissues. All of these are basically substances that consist of N-acetylglucosamine in its polymer form. Glycoproteins are another interesting topic. Glycoproteins are really just what happens when you use glucose and glucose derivatives as building blocks for larger structures. And one of the places it's most relevant in the health sciences is glycoproteins are what give us cell surface receptors. So the different blood types that human beings can have, whether it's A, B, O, or AB, all are just referring to different combinations of glycoproteins, which are really just glucose derivatives on the surface of those red blood cells. And in fact, if you know your blood type, you can very quickly determine exactly what glycoproteins are attached to your blood cells. For example, if you have type O blood, it would have the sugar fucose chemically bonded to galactose and in turn bonded to N-acetylglucosamine. And you can see, I'll leave this up for a second, depending on what your blood type is, we just have different arrangements of these different structures. All of these are based on either a monosaccharide sugar or a derivative of a monosaccharide sugar, and then they're linked together. Again, the main thing I want you to remember is just that you can take these monosaccharide structures, combine them together to make disaccharides, or combine thousands of them together to make polysaccharides. If you go on to Chemistry 123 or any other chemistry course, you'll start to see these in more detail. But that's where we'll end for our topic of carbohydrates. If you have any questions, please let me know.